Hello everyone, my name is Xingfen Shao from University of Southern California. Today I will present our work about high-resolution imaging of choroid plexus blood flow using ASL. And I have nothing to disclose. Choroid plexus presents in each of the four ventricles and produces and regulates most of the CSF in the central nervous system. CSF serves as a drainage pathway for the brain metabolic waste and toxins as part of the lymphatic system. So study of the choroid plexus blood flow could provide insights into brain clearance mechanisms and related pathologies such as Alzheimer's disease. Recently, ASL has been proposed to measure choroid plexus blood flow non-invasively. So the goal of this study is to develop an ASL sequence with higher spatial and temporal resolution and to propose a post-processing denoising method to enhance dynamic choroid plexus ASL signal. So here's our protocol. High-resolution ASL data was acquired with 2 mm isotropic resolution and with 24 slices to cover all ventricles, as indicated by blue boxes here, and with 15 post-lesion delays ranging from 500 to 2700 milliseconds to capture the dynamics of perfusion signal. We have scanned 4 healthy subjects and 1 normal pressure hydrocephalus patient. Choroid plexus was segmented from T1 MP ridge using etiquette snap toolbox, as indicated by red arrows. So here shows center 20 slices of perfusion images, averaged from 15 post-lesion delays, and overlaid with choroid plexus mask, as shown in red color. And here shows 4 zoom slices with regions of choroid plexus indicated by red arrows. Strong choroid plexus blood flow signal can be observed and match well with a mask segmented from structure images. We utilize the post-processing method to further improve the ISMRM of dynamic perfusion signals, called k-space weighted image average or QIA. This method generates k-space as a weighted average of high-frequency k-space signal between adjacent time frames, and can nicely preserve both spatial and temporal resolution. For more details, please refer to this oral presentation by my colleagues from USC. So here, the top row shows the center slice of original dynamic perfusion images and 15 post-lesion delays as shown from left to right. And the bottom row shows the perfusion images after QIA reconstruction. High-quality dynamic perfusion images were obtained with only 1 pair of label control and each PLD. And QIA reconstruction further enhanced the ISMRM without compromising the spatial or temporal resolution. A dynamic perfusion change in choroid plexus can be easily observed, as indicated by these red arrows. So here are the structure image and the perfusion map from 1 healthy subject and 1 normal pressure hydrocephalus patient. Enlarged ventricle as well as increased choroid plexus volume can be observed in this NPH patient, as indicated by those 2 red arrows. Here are the scatter plots of average perfusion signal and fitted curve in gray matter and choroid plexus, average across 4 healthy subjects on top, and in the NPH patients, the bottom. Fitting the dynamic perfusion signal to this ASL model, T1, arterial transit time, and blood flow can be computed simultaneously. So blood flow in gray matter and choroid plexus were comparable in healthy subjects, and reduced blood flow was observed in this NPH patient. It is worth noting here, choroid plexus flow was about 30% higher than that of gray matter in this NPH patient, suggesting that relative high choroid plexus blood flow might be associated with CSF overproduction in NPH. In conclusion, we propose a high-resolution multi-delay pCASL sequence and a QIA denoising algorithm to reliably measure dynamic blood flow signal in choroid plexus, and choroid plexus blood flow may be altered in NPH patients. We also observe a potential trend between water exchange rate across a blood-brain barrier, Kw, measured by a diffusion-prepared ASL sequence. We found a more positive association between choroid plexus blood flow and Kw than blood flow in gray matter, and a negative trend between choroid plexus blood volume and Kw, which indicates water exchange across the BBB may be associated with CSF regulation and lymphatic system. Thank you for your attention.